Thank you for coming. My name is Sofia Fertuzinhos, and today I will share with you how the Bioinformatics Support Hub has been bridging the bioinformatics knowledge gap by biomedical researchers through facilitating access to omics data analysis tool. The generation, analysis, and interpretation of omics data have become indispensable in modern biological research. However, many researchers, particularly those in advanced stages of their careers, lack the computational skills necessary for this analysis. Adding to the lack of computational skill, researchers must deal with constant technological innovation accompanied by new analytical methodologies. While bioinformatics services are increasingly offered by core facilities, the access to them is limited in terms of costs, as junior researchers tend to have less funds to afford them. And in terms of effective communication of the results in language, researchers can fully grasp. So the Bioinformatics Support Hub is a group from the Yale Cushing Whitney Medical Library and is led by two librarians, my colleague Rolando Garcia-Milian and myself. The mission of the hub is to meet researchers at their level of bioinformatics needs and empower them with the knowledge, skills, and tools necessary so they can advance their research as in terms of publishing more, impacting their clinical practice, and getting more funding. The hub has developed a program to address the need for bioinformatics through a defined cycle of omics data analysis, beginning at data downloading, step 1, from experimental generated data from Yale sequencing core or public repositories, followed by a step 2, which is processing this raw sequencing data. Then researchers will want to perform some statistical analysis, after which they may want also to perform pathway analysis to understand the biological effects of their experimental manipulations, which is step 4, or further narrow their analysis to produce an hypothesis that can be submitted either for publication or as grant proposal. And finally, researchers may need to upload their omics data to a public repository to comply with funding agency or journal requirements, and that's step 6. The hub strategy to assist researchers in all of these steps lays on 4 approaches. The first one, promote access to user-friendly bioinformatics licensed software, such as Partec Flow, to aid on data processing, ClueCore Omics Explorer to aid on statistical analysis and visualization, and IPA and Metacore for pathway analysis. The next focus is in organizing workshops on how to use public resources, mostly public resources that can be delivered by us, that are delivered by us in the hub or by the community members or peers, as well as by guest speakers, such as core leaders of internal resources. I will talk about these workshops in more details shortly. Third, we provide personalized consultations to address specific questions by researchers on their projects. And last, we are available to perform specific data analysis for researchers through project collaborations. The impact of this strategy is reflected in this past year's numbers of attendees to our workshops, 622 out of 1,422 registrants. For one-on-one consultations, we met 240 times with 186 patrons, and we are currently collaborating in 20 projects and have been included in the authorship of 8 accepted publications just in the last year. So in more detail, the number of attendees to our workshops are split among several strategies of teaching. So the first one is our workshops organized by the Bioinformatics Support Hub that are delivered by Rolando and I. These are one-off standalone workshops. And we try to create the habit to be always on the same day and for the maximum of 2 hours. So this past year was Bioinformatics on Thursdays. And we delivered in this past year 17 hands-on workshops that had 340 attendees out of 813 registrants. And on average, you can see in this histogram here, 20 attendees per session. These workshops aim to teach on how to use different public softwares, such as gene set enrichment analysis or Cytoscape to perform pathway annotations, and how to run R packages such as dSeq2 to perform differential gene expression, bulk RNA-seq, and BioMAR to perform gene annotation. These 17 hands-on workshops, 14 of them were in person and 3 were online. These workshops are well received and I would say well participated. And when asked, participants don't shy away from asking for more and more diverse topics, which are hard to satisfy just by the hub. So, for example, these participants would like to have more epigenetic data analysis. These others asked for single cell analysis. Some asked for more biostatistics topics, and also how to access to computers on clusters. So, which are actually provided by cores at Yale. So to respond to these needs, we have actually reached out to the community, and peer-to-peer teaching has been successfully implemented to address these knowledge gaps. So peer teachers have the advantage of being more likely aware of the current challenges faced by their peers when trying to learn the same skills. In the past year, we have organized the presentation of 14 peer-to-peer workshops that had 176 attendees out of 378 registrants. The teaching subjects are mostly on different omics data analysis using R and Python, but not only. We also have promoted classes on image analysis methodologies and statistical analysis and experimental design and science communication. These peer-to-peer sessions are also standalone. However, with the presenter's permission, we record the workshops so they can be seen asynchronously by people that attended the sessions but need more, need to review it and to be more clear, or people that failed to attend the sessions and would like to see it asynchronously. Additionally to the standalone workshops, we have also delivered a teaching series of workshops that guided participants from basic concepts of genomic data annotations to where to find public available data, specifically bulk RNA-seq data, that we showed how to download and import into RStudio and then analyze with DSeq2 for differentially gene expression analysis, then how to add gene annotations with Biomart, and followed by visualizations of results with ClueCore, and finally 2 sessions about 2 different approaches to perform pathway analysis, such as overrepresentation analysis and gene set enrichment analysis. These 7 sessions were spread over 1.5 months, 2 sessions per week of 2 hours duration maximum. 2 of these sessions were delivered by guests that presented their classes online. Interestingly, even though more participants were allowed to attend the online sessions, participation was around 50%, which was very similar to what we observed in in-person sessions. In total, over 100 attendees came to our workshops, and out of 206 registrants that could come to all. So these attendees could come to all sessions or just to some or one workshop according to their interests. Last, on the teaching front, we organized presentations from leaders of core units on their services or invite researchers to speak about how the community can take advantage of internal resources. These are normally of wider interest and so well attended. To give you an example, we invited Dr. Cohen, director of psoriasis treatment program, to share how to use the database All of Us, which is a national project that aims to collect genetic and health data from 1 million volunteers. And so we invited him to share with the community how he used this database to study genetic variations associated with skin pathologies. Also, we invited leaders, of course, such as those responsible for sequencing, like the YCGA, the Yale Center for Genomic Analysis, to present together with the Yale Center of Research Computing. So researchers understand how these 2 units work together to make their data available and manageable, so they can use the course, the clusters as one of the participants in our workshop suggested, and use them to analyze bigger data sets. So overall, teaching workshops and seminars are attended by a diverse population of researchers, but who seems to make most advances are postdocs, followed by graduate students, followed by others, such as clinicians, and faculty and associated research scientists, and lastly, supporting staff. As I have mentioned, a natural sequence to the workshops is the request for one-on-one consultations to address the researcher's specific needs. And like workshops, these consultations are free and unlimited. Well, these are unlimited. Workshops are standalone and they have a different format. But consultations serve to explore methodologies and approaches to omics data analysis specifically for the researcher, as well to solve problems related to licensed or public softwares that they have learned how to use, or an R script that they have received and they cannot make it work, or how to arrange data wrangling, so how to deal with data that and clean it so they can analyze it. In terms of impact, a recent survey to people that received consultations revealed that these consultations helped researchers mostly on ongoing projects. And that's why they come to us multiple times throughout their project, but also preparing results to present at meetings. And for grant applications and and importantly, to publish a manuscript, develop thesis work. And a few people responded other. I wanted to share this one that filled my heart when these researchers said that the hub had helped convince hers or his PI that the data was worth publishing. Most researchers benefiting from consultation hold a postdoc position, followed by associate research scientists, so very similar to what we see in workshops. And interestingly, among faculty, assistant professors who are beginning their tenure track and have less resources have more advantage had taken more advantage of the service provided by the hub than others more senior faculty. In a recent survey, the hubs received high ratings, and it was praised for their helpfulness, patience, knowledge, and availability, and for their willingness to explain concepts multiple times to ensure their understanding. Understandably, there is always room for improvement, and one person expressed that they would like to receive more specific input on their project. In addition to consultation, some researchers, such as principal investigators and faculty, entrust the Bioinformatics Support Hub with data analysis of sections of future projects by writing them into a grant application or an ongoing project. This requires a deeper understanding of the researcher's project, including hypothesis, experimental design, and expected results, leading to highly interactive meetings. Currently, the hub is involved in 20 ongoing projects, and last year we had been included in the authorship of 8 publications. In this network analysis, we show the number of departments with which the hub has published since its inception in 2014, in a total of 40 publications to date. In summary, the Bioinformatics Support Team in the Yale's Cushing's Whitney Medical Library acts as a glue unit facilitating the communication between core facilities and researchers, and importantly, has developed a program that seeks to meet the ever-evolving bioinformatics needs of users while fostering a culture of learning and collaboration within the Yale biomedical community. Thank you for your interest and attention. And if you have any questions, don't hesitate to reach me at sofia.fertuzinhos@yale.edu. Thank you again.